first comprehensive network of wild crop species will help breeders tackle food insecurity. The first comprehensive network of sites where crop wild relatives are found has been developed by researchers at the University of Birmingham. The network will help breeders develop more resilient crops and tackle challenges of global food security, as well as being important for nature conservation. The resource includes details of the 150 hotspot sites worldwide where some 1,261 so-called crop wild relative (CWR) species are concentrated. Crop wild relatives are cousins of cultivated crops and a vast resource of genetic diversity. Plant breeders will be able to use the diversity conserved within these sites to transfer adaptive traits from the wild relatives to domestic crops to produce new varieties that will produce higher yields and are more resistant to climate change or pests. The sites identified by the Birmingham team are mostly within existing conservation areas, which will help to ensure these valuable wild species can be conserved well into the future. They contain relatives of 167 of the most important crops in the world as listed by the UN Food and Agriculture Organization (FAO). Identifying the 150 sites will mean the species can be conserved in situ, to complement and extend the diversity already held in ex-situ gene banks, which cannot alone preserve the full range of diversity found in nature. Interestingly, the most hotspots identified were in the so-called Fertile Crescent, which includes countries in the Middle East such as Lebanon, Egypt, Syria and Turkey, the area of the world where modern-day agriculture first began. Ensuring global food security is one of the greatest challenges of our time as population growth puts ever more pressure on our food production and natural ecosystems, explains senior author Dr. Nigel Maxted, of the University of Birmingham's School of Biosciences. Finding new, more resilient crop varieties that can withstand climatic extremes, be more resistant to pests or require less water to thrive is a huge priority for plant breeders. To find that diversity, you need to go back to the ancestral species, the wild crop relatives, from which our crops were domesticated. The resource we have developed will enable breeders to identify where to obtain the necessary crop traits from which to breed vital new climate-smart varieties. Having identified the key 150 global populations, the next step for the team will be to develop a unifying management network, through the FAO, to manage these sites to sustain the genetic diversity of CWR populations for the future benefit of humankind. The research was published in Nature Communications Biology, and was funded by a grant from the Government of Norway via the Global Crop Diversity Trust. Project partners include the Global Crop Diversity Trust, the Royal Botanic Gardens Kew and the International Centre for Tropical Agriculture. Story source. Materials provided by University of Birmingham. Note: content may be edited for style and length. Journal reference. Holly Vincent, Ahmed Amri, Nora P. Castañeda Alvarez, Hannes Dempuwolf, Asan Delu, Luigi Guarino, David Hole, Chikalu MBA, Alvaro Toledo, Nigel Maxted. Modeling of crop wild relative species identifies areas globally for in situ conservation. Communications Biology, 2019, 2-1, doi, 10.1038 per seconds 42003-0190372Z. Eastern forests shaped more by Native Americans burning than climate change. Native Americans' use of fire to manage vegetation in what is now the eastern United States was more profound than previously believed, according to a Penn State researcher who determined that forest composition change in the region was caused more by land use than climate change. I believe Native Americans were excellent vegetation managers and we can learn a lot from them about how to best manage forests of the U.S., said Mark Abrams, professor of forest ecology and physiology in the College of Agricultural Sciences. Native Americans knew that to regenerate plant species that they wanted for food, and to feed game animals they relied on, they needed to burn the forest understory regularly. Over the last 2,000 years at least, according to Abrams, who for 3 decades has been studying past and present qualities of eastern U.S. forests, frequent and widespread human-caused fire resulted in the predominance of fire-adapted tree species. And in the time since burning has been curtailed, forests are changing, with species such as oak, hickory and pine losing ground. The debate about whether forest composition has been largely determined by land use or climate continues, but a new study strongly suggests anthropogenic fire has been the major driver of forest change in the East, said Abrams. That is important to know because climate change is taking on an ever larger proportion of scientific endeavor. But this phenomenon does not apply to other regions, Abrams noted. In the western U.S., for example, climate change has been much more pronounced than in the East. That region has received much more warming and much more drought, he explained. Here in the East, we have had a slight increase in precipitation that has ameliorated the warming, said Abrams. To learn the drivers of forest change, researchers used a novel approach, analyzing both pollen and charcoal fossil records along with tree census studies to compare historic and modern tree composition in the forests of eastern North America. They looked at 7 forest types in the north and central regions of the eastern United States. Those forest types encompass 2 distinct floristic zones, conifer northern hardwood and sub-boreal to the north, and oak pine to the south. The researchers found that in the northernmost forests, present-day pollen and tree survey data revealed significant declines in beech, pine, hemlock and larches, and increases in maple, poplar, ash, oak and fir. In forests to the south, both witness tree and pollen records pointed to historic oak and pine domination, with declines in oak and chestnut and increases in maple and birch, based on present-day data. Modern forests are dominated by tree species that are increasingly cool-adapted, shade-tolerant, drought-intolerant pyrophobes, trees that are reduced when exposed to repeated forest burning, Abrams said. Species such as oak are largely promoted by low to moderate-level forest fires. Furthermore, this change in forest composition is making eastern forests more vulnerable to future fire and drought. Researchers also included human population data for the region, going back 2,000 years, to bolster their findings, which recently were published in the Annals of Forest Science. After hundreds of years of fairly stable levels of fire caused by relatively low numbers of Native Americans in the region, they report, the most significant escalation in burning followed the dramatic increase in human population associated with European settlement prior to the early 20th century. Moreover, it appears that low numbers of Native Americans were capable of burning large areas of the eastern U.S. and did so repeatedly. After 1940, they found, fire suppression was an ecologically transformative event in all forests. Our analysis identifies multiple instances in which fire and vegetation changes were likely driven by shifts in human population and land use beyond those expected from climate alone. Abrams said. After Smokey Bear came on the scene, fire was mostly shut down throughout the U.S. and we have been paying a big price for that in terms of forest change. We went from a moderate amount of fire to too much fire to near zero fire, and we need to get back to that middle ground in terms of our vegetation management. Also involved in the research was Gregory J. Nowacki, with the Eastern Regional Office, U.S. Department of Agriculture Forest Service. The Agricultural Experiment Station of Penn State funded this research. Ammonium fertilized early life on Earth. A team of international scientists, including researchers at the University of St Andrews, Syracuse University and Royal Holloway, University of London, has demonstrated a new source of food for early life on the planet. Life on Earth relies on the availability of critical elements such as nitrogen and phosphorus. These nutrient elements are ubiquitous to all life, as they are required for the formation of DNA, the blueprints of life, and proteins, the machinery. They are originally sourced from rocks and the atmosphere, so their availability to life has fluctuated alongside significant changes in the chemistry of Earth's surface environments over geologic time. The research, published in Nature Geoscience, reveals how the supply of these elements directly impacted the growth of Earth's oxygen-rich atmosphere and were key to the evolution of early life on Earth. The most dramatic change in Earth history followed the evolution of oxygenic photosynthesis, which fundamentally transformed the planet by providing a source of carbon to the biosphere and a source of oxygen to the atmosphere, the latter culminating in the Great Oxidation Event, GO, some 2.3 billion years ago. Despite the critical importance of nutrients to life, the availability of nitrogen and phosphorus in pre-GO oceans is not well understood, particularly how the supply of these elements drove and/or responded to planetary oxygenation. Using samples of exceptionally well-preserved rocks that have been associated with early evidence for oxygenic photosynthesis 2.7 billion years ago, the team of researchers examined Earth's early nitrogen cycle to decipher feedbacks associated with the initial stages of planetary oxygenation. There is precious little rock available from this time interval that is suitable for the type of analyses we performed. Most rocks that are this old have been deformed and heated during 2.7 billion years of plate tectonic activity, rendering the original signals of life lost, says Christopher Junium, Associate Professor of Earth Sciences in the College of Arts and Sciences. The rock samples showed the first direct evidence of the buildup of a large pool of ammonium in the pre-Go oceans. This ammonium would have provided an ample source of nitrogen to fuel the early biosphere and associated oxygen production. Research team leader Aubrey Zirkle, reader in the School of Earth and Environmental Sciences at the University of St. Andrews, says, "Today we think of ammonium as the unpleasant odor in our cleaning supplies, but it would have served as an all-you-can-eat buffet for the first oxygen-generating organisms, a significant improvement on the dumpster scraps they relied on earlier in Earth's history." As well as helping scientists better understand the role of the nitrogen cycle in global oxygenation, the new findings also provide context for other nutrient feedbacks during early planetary evolution. It is becoming ever more clear that the game of nutrient limitation has tipped back and forth through Earth's history as life has evolved and as conditions have changed, Junium says. Surprisingly, evidence for significant atmospheric oxygenation does not appear until 400 million years later, meaning that some other nutrient, such as phosphorus, must have been important in setting the evolutionary pace. Extreme draining of reservoir aids young salmon and eliminates invasive fish. A new study finds that the low-cost, extreme draining of a reservoir in Oregon aided downstream migration of juvenile Chinook salmon, and led to the gradual disappearance of 2 species of predatory invasive fish in the artificial lake. The study is published in the journal Ecohydrology. The elimination of largemouth bass and crappie from Fall Creek Reservoir, about 30 miles southeast of Eugene in the Willamette River Basin, could have management implications for reservoirs that have been invaded by certain species of fish that eat other fish, according to Christina Murphy, a recent Oregon State University doctoral graduate and lead author on the study. Even though the strategy appears extreme, it's both helping juvenile salmonids pass downstream and promoting a native species-dominated reservoir, Murphy said. Bass and crappie, which are major predators in the reservoir, have been pushed out and into the Willamette. That can be worrying because filling the river with invasive, warm water fish seems like a bad idea, but these reservoirs are leaking warm water fish all the time. The good news is that they were pushed downstream in the fall and winter, when the water is cold and the flow is faster, so they should be at a disadvantage in the Willamette River, compared to when they leak out of the reservoir in the summer. Fish passage out of reservoirs is a critical issue for downstream movement of juvenile salmonids and other migratory species. Because of their declining numbers through the decades, 9 West Coast populations of Chinook salmon are federally threatened and endangered species. In the Pacific Northwest, large dams and reservoirs have been constructed on many salmon-bearing rivers. Salmon are anadromous, they migrate from home streams to the ocean as juveniles and return a few years later as adults to spawn, so damming rivers can delay downstream migrations by juvenile salmon for months or years. For the past several years, the U.S. Army Corps of Engineers has briefly drained Fall Creek Reservoir in the fall to help juvenile spring Chinook salmon move out of the reservoir and continue their downstream migration. A team of researchers from Oregon State, the U.S. Forest Service Pacific Northwest Research Station and the Corps of Engineers analyzed fish capture data from 2006 to 2017. Annual draining started halfway through this period, to examine changes in timing of passage out of the lake and to compare fish species composition. The team found that draining the 50-year-old reservoir led to its intended result, giving juvenile Chinook salmon a way out of the reservoir. At the same time, crappie and largemouth bass numbers began declining until there were none. We have also been sampling fish within the reservoir during the summer, Murphy said. In 2012, we could capture 10 bass an hour. This went down each year. By the summer of 2015 we only caught 1 during all of our sampling, and in 2016, we didn't capture any. This change was reflected in the data from the trap downstream too. The decline occurred with crappie, but faster. This extreme draining management is only about a week in duration, but has implications for the whole ecosystem. It makes the reservoir begin to act as a natural river again, affecting the entire fish community, said Ivan Arizmendi, an assistant professor in the Department of Fisheries and Wildlife in OSU's College of Agricultural Sciences and co-author on the study. Sherry Johnson, a research ecologist with the U.S. Forest Service Pacific Northwest Research Station, said, "This type of research and monitoring before and after major changes in management of a reservoir is crucial for improving our ability to balance water availability while maintaining native species." Gregory Taylor and Todd Pierce of the U.S. Army Corps of Engineers were co-authors on the study. Funding for the research was provided by the U.S. Army Corps of Engineers. Ecological factors influences the distribution of lionfish on deep reefs. Invasive lionfish, Pterois volitans and P. miles, are now ubiquitous throughout the Caribbean and Western Atlantic on both shallow and deep reefs. While many invasive species disrupt natural ecosystems by spreading disease or competing for food and habitat, lionfish are particularly problematic owing to their voracious appetites and high reproductive capacities. Lionfish are indiscriminate predators, feeding on over 70 species of fish, invertebrates, and mollusks, and a single lionfish can eat 30 times its stomach volume in one meal. Additionally, each female lionfish can produce more than 2 million eggs each year during her average 16-year lifespan, making lionfish a significant threat to native fishes and reef systems. Recent surveys in Bermuda have revealed dense aggregations of lionfish on mesophotic reefs, located at depths between 100 and 500 feet, or 30 to 150 meters, yet these densities are not consistent across reefs at this depth. Newly published research in the journal Frontiers, led by BIOS reef ecologist Gretchen Goodbody-Gringley, examines how lionfish distribution on mesophotic reefs may be affected by ecological factors, such as the abundance of prey organisms, as well as environmental factors, such as water temperature. The research team, which included scientists from the University of Massachusetts at Dartmouth, the Bermuda Department of Environmental and Natural Resources, and the Bermuda Natural History Museum, used diver-led visual surveys to investigate 11 sites at a depth of 196 feet 60 meters. During the surveys, divers recorded numbers of both lionfish and prey fish, as well as the size class of individual prey fish and the bottom temperature. High densities of lionfish were recorded in areas with higher abundances of prey fish, specifically the Creole fish Parantheas furcifer, which is a preferred prey among lionfish, and higher prey fish biomass. However, the influence of seawater temperature was found to have the strongest effect on lionfish distribution, where higher lionfish densities were found at sites with lower bottom temperature. Lower temperatures also were correlated with higher densities of prey fish biomass, and P. furcifer biomass, implying that physical factors in the environment, such as temperature, likely influence the ecology of the same area. These results suggest that cold water upwelling currents may be fueling the food chain in certain locations, resulting in high abundances of prey fish and, thus, lionfish. Understanding why high densities of lionfish occur in specific locations will aid the ability of scientists and managers to identify and target areas for lionfish control efforts, thereby increasing the efficacy of future management. Goodbody-Gringley said. Story source. Materials provided by Bermuda Institute of Ocean Sciences. Note: content may be edited for style and length.